Hello, and welcome to the Integrated DNA Technologies webinar titled Development of Digital PCR-Based Products to Test for Cancer-Related Gene Mutations in Cancer Tissue and Liquid Biopsies. My name is Francisco Klein, and I am a Senior Commercial Product Manager at IDT. I'll be serving as the moderator for today's presentation. This presentation will be given by Dr. Seung-Hee Hung. Dr. Hung is the medical science liaison at Gencurix. She received her PhD in cancer biology from Seoul National University. Following her doctoral studies, Dr. Hung spent 15 years as a researcher at the National Cancer Center in Korea. And later, she spent 2 years as a postdoctoral fellow at the National Institutes of Health in the United States. Throughout her career, she's concentrated her research in the areas of lung, colon, breast, and pancreatic cancers. This presentation should last about 40 minutes. Upon completion, I'll lead a question and answer session where Dr. Hung will address as many questions as possible. As attendees, your microphones have been muted, but we encourage you to ask questions or make comments at any time during or after the presentation by entering your questions in the QA box at the bottom of the screen. Also, please note that you can expand the presentation slide window for easier viewing. In case you need to leave early or want to revisit this webinar, we are recording the presentation and will make a link to the recording available on our website in a few days. You will receive this link in a follow-up email. Now, enjoy the presentation.
Thank you, Francisco, for the introduction. Hello, everyone. My name is Seung-Hee Hong. I'm the Medical Science Liaison at the Gencurix. Today, I will be presenting on the topic development of digital PCR-based product to test for cancer-related gene mutations in cancer tissue and liquid biopsies. Here's the table of contents for presentation. First, I'd like to explain digital PCR. The first generation of conventional PCR was a method that allowed qualitative analysis of genes through agar-agar gel electrophoresis, but quantitative analysis was difficult. To complement this, the second generation of real-time PCR was developed, which allows quantitative and qualitative analysis of gene expression through relative quantitative analysis. However, real-time PCR has the disadvantage that a standard curve is required for absolute quantitative analysis, and the result may vary depending on the efficiency of the PCR. To overcome this, the concept of third-generation digital PCR has been applied. Digital PCR does not require the process of drawing a standard curve and can be analyzed in absolute values. Sample partitioning is the key to digital PCR. In the DrumNet digital PCR system, a single PCR sample is partitioned into 20,000 drumnets. Template molecules are distributed randomly among drumnets, such that some drumnets have no template molecules and others have one or more. Each drumnet undergoes PCR amplification and analysis separately, and the drumnet are then individually counted and scored as positive or negative for fluorescence. This process is known to be possible in 1 day. Also, the main advantage of digital PCR is its good sensitivity. As you can see from the graph, the advantage of digital PCR over qPCR or NGS is in sensitivity, specificity, cost effectiveness, and turnaround time were well established. In addition to Bio-Rad best known two-color labeled QX200, six-color labeled QX600 has been developed. And other competitors have also introduced multiplexable digital PCR instrument. We expect the digital PCR market to grow significantly in the future. Now I'm going to start talking about digital PCR as it is used in cancer research. In the figure to the left, you can see the difference in sensitivity for each test method based on single sequencing. Digital PCR can detect variants at the lower sensitivity compared to real-time PCR and standard NGS. It also has a significant cost advantage over NGS in case where a known genetic variant needs to be confirmed for drug prescriptions or monitoring. The use of the PCR in cancer research has increased in recent years for the following key reasons. The high sensitivity and accuracy of this PCR make it highly favorable for detecting extremely low concentration of genetic or rare variants. This is particularly advantageous for detecting circulating tumor DNA in the blood from liquid biopsy. For this reason, it is being utilized across cancer research, increasing early cancer detection, MLD, treatment selection, and monitoring of drug response. Next time, talking about detecting mutations in cancer tissue liquid biopsy. I will now explain the background to study of each of the genes known as companion diagnosis, prognosis diagnosis, and drug resistant diagnosis for each type of cancer and how they can be tested using this PCR. The first is about testing for EGFR gene mutations in non-small cell lung cancer. The year 2024 marks 20 years since the discovery of activating mutations in the EGFR in NSCLC. Since then, there have been major improvements in treating NSCLC based on this discovery. Some of these breakthroughs have caused a significant shift in how cancer research is understood and have driven process in treatment beyond the NSCLC. The figure depicts a timeline describing the generational evaluation of EGFR tyrosine kinase inhibitors. EGFR TKI are important therapies in the treatment of non-small cell lung cancer. This shows how each generation of drugs target EGFR mutations with increasing efficacy and the duration of treatment over time. In recently years, almost all tumor type and even plasma samples from patients with NSCLC have been routinely molecularly profiled for multiple actionable driver mutations, and profiles is not limited to advanced refractory stages. In addition, plasma genotyping is increasingly being used to monitor treatment responses and detect resistance to targeted therapies. In addition, a number of studies have used highly sensitive digital PCR-based assays to detect EGFR gene mutations in plasma. A number of studies have confirmed the advantage of the dPCR, especially when compared to NGS, such as concordance with NGS and faster TAT. GenQuix provides molecular diagnostic solutions across the cancer care continuum. Droplex lines are in the IVD kits that detect genes important for the diagnosis for non-small cell lung cancer, endometrial cancer, colorectal cancer, breast cancer, etc. Droplex EGFR mutation test version 2 detects a total of 107 mutations in EGFR, which is more than twice the number detected by the competing products. The droplex EGFR mutation version 2 can be detected on FFP and plasma specimen and has received the CIVD approval and the IVD from the Korea FDA and currently preparing for IVDR. These tests are especially targeted for type of EGFR gene exons 18, 19, 20, 21. Additionally, it has the capability to identify various targeted anti-cancer drug resistance mutations, especially C797S or G, which is known to be associated with resistance to osimertinib. It has more variants of exon 19 deletion and exon 20 insertions than other products. We believe it has the highest EGFR mutations coverage of any PCR-based product. Next, I will talk about polygene mutation testing, which has recently been recommended for endometrial cancer. Cancer of the corpus luteally is usually referred to as endometrial cancer. The global incidence of endometrial cancer continues to rise, especially in young women. The cancer is particularly prevalent in high-income countries such as North America, Eastern Europe, and Northern Europe. One of the main factors contributing to the rising incidence of endometrial cancer is the rising obesity rate worldwide. The second reason is reproductive factors. In 2013, the TCGA group performed the exome sequencing on the total of 248 paired endometrial tumor and normal samples. The purpose of the study was to determine if a more accurate molecular diagnosis could be made using NGS rather than histological diagnosis, which was previously divided into two groups. As a result, they found that patients could be divided into 4 subgroups by mutation types as follows. And among them, patients with polymutations are more likely to be ultra-mutated and have a good prognosis. Major milestones in endometrial cancer clinical practice have led to the following outcomes. Significant change in the treatment of polymutations endometrial cancer. Following the publication of the TCGA result in 2013, testing for polygene mutation was included in the ESMO guidelines in 2016. And an additional study, the PROMISE classifier, was published in 2017 to provide a practical way to test for poly mutations. In addition, the PORTEX-3 study in 2018 confirmed that endometrial cancer patients with a polygene mutation have a good prognosis. Based on these studies, the need for polygene mutation testing has since been empathized by the various international cancer organizations and updated on their guidelines. Patients with polygene mutation account for 5 to 15% of all endometrial cancer patients. They are mostly younger women with high-grade endometrial disease. Most variants are characterized by high prevalence of the five hotspots and a good prognosis. Several clinical trials are now on the way to determine whether diagnosing these polygene mutations can help tailor certain treatment strategies. In the future, positive results from this trial may allow patients with poly mutations to skip standard chemotherapy, reducing the unnecessary over treatment and helping patients maintain their quality of life. If this is the case, the poly test may be the first essential test. Recently, it has been confirmed that poly mutations induce a hypermutation state, which increases the mutation burden. This means that more neoantigens are produced, which are more likely to elicit an immune response and may be more responsive to immunotherapy. As a result, immunotherapies such as immune checkpoint inhibitors are more likely to be effective. Therefore, studies are on the way to determine if patients with polymutant endometrial cancer may be good candidates for immune checkpoint inhibition. Previously, polygene mutations were tested by NGS and Sanger test method. Here I explain the advantage and disadvantage of NGS, Sanger, and dPCR. In the case of NGS, all polymutations can be identified, but not just the hotspot. And the sensitivity is high, but the reality, the test price is expensive and the TAT is long. For Sanger sequencing, primer design allows you to test for all polyvariants and it's economical. But its low sensitivity can lead to inaccuracies in the analysis of result and requires a large amount of sample for testing. The dPCR cannot see all polymutations, but it can test for hotspot mutations listed in the guidelines. It is relatively inexpensive compared to NGS, and TAT is faster and more sensitive and making it more accurate. Based on this dPCR-based technology, Gencri's droplex polymutation test was launched, received FDA approval in Korea last year, and has been used and published in several various research papers. We have already received CIVD for Europe and are currently in the process of receiving CIVDR. Finally, I'd like to introduce the latest trend in ESR1 mutation testing in ctDNA from HR positive or HER2 negative metastatic breast cancer patient. And the droplex is ESR1 mutation test kit, which is currently being used in several national and international studies. Breast cancer is the most common cancer in women worldwide, with hormone receptor positive breast cancer accounting for the significant proportion. While a variety of therapies are available to treat these patients after surgery, endocrine therapy is often used as a first-line treatment for HR-positive breast cancer. In addition, the combination of CDK4 and 6 inhibitors and endocrine therapy has recently become the standard of care for first-line treatment of HR-positive and HER2-negative advanced or the metastatic breast cancer. And the selection of second-line treatment for recurrence after first treatment, different biomarkers are being tested to select a different treatment for each gene mutations. In other words, the testing for biomarkers have become a necessity in precision medicine for personalized treatment selection for cancer patient. The endocrine therapies of choice for first-line treatment of HR-positive patients include aromatase inhibitors, SOMs, and SERD and PROTECts. While the mechanism of action of each drug is different, they all have the same goal of inhibiting cell growth by interfering with the function of estrogen and estrogen receptors. These drugs play an important role in making breast cancer treatment strategies more personalized and effective. However, while most HR-positive breast cancer responds to first-line endocrine therapy for 1 to 4 years, many patients eventually develop resistance, posing the significant challenge in patient management. ESR mutations are known to be a major mechanism of endocrine drug resistance in HR-positive breast cancer. Normally, therapeutics bound to estrogen ligand work through multiple mechanisms to inhibit cancer growth. However, ESR mutations alter the structure of ligand binding domain of DER, reducing the affinity of DER for estrogen, SOMs, and SERD, and enhancing growth, cancer growth, metastasis, and resistant mechanisms. Almost all ESR mutations were found in codons 310 to 547. Among them, 4 hotspot mutations account for about 3/4 of all ESR1 acquired mutations. Several studies have shown that ESR mutations are rarely found in patients with early breast cancer who are not receiving endocrine therapy. However, in patients with advanced or metastatic breast cancer who have been treated with endocrine therapy, ESR mutations are found in 10% of patients on the first-line therapy and 40% of second-line therapy and more than 50% on third-line therapy and resistant develop. Thus, in recent years, several studies have shown that in patients prescribed endocrine therapy, testing for variant of ESR1 can be used to quickly determine if patient is resistant by checking for variants at different stages of treatment. In addition, a paper published this year reported that the breast cancer patient, especially for the ESR gene, testing concurrently with blood samples rather than tissue alone can detect more actionable mutations. Based on these studies, several guidelines have recently been published regarding testing for ESR mutation gene mutations in patients with HR positive and HER2 negative advanced or metastatic breast cancer. For asthma guidelines, recommended testing for ESR mutations in ctDNA samples as a priority and noted that in patients with advanced breast cancer receiving aromatase inhibitors and CDK4 and 6 inhibitor therapy, monitoring for ESR mutations is likely to be clinically useful. The 2023 ASCO guideline also recommended routine testing for the emergence of ESR mutations in patients with HR positive HER2 negative breast cancer at the time of recurrence or the progressions during endocrine therapy to help guide the treatment selection. Finally, the recently updated NCCN guidelines state that ESR mutations should be tested for the patient with disease progressions after endocrine therapy using NGS and PCR-based tests in tissue or blood specimens, and that a new FDA-approved elacestrant should be prescribed if ESR mutations are found. In summary, for HR positive, HER2 negative, advanced metastatic breast cancer patient with previously undetected ESR mutations, routine ctDNA testing is recommended as a first-line screening at any stage of disease progressions. So, what are different methods for testing for ESR1 gene mutations? As we have shown, most clinical studies have been based on NGS and drug-net distal PCR. Since then, a number of clinical studies have been published. To summarize the PADA-1 trial, it tested the clinical efficacy and monitoring ESR1 mutations in ctDNA using drug-net distal PCR to guide an early switch from AI plus palbociclib to fulvestrant plus palbociclib. The trial shows that patients with ESR mutations had a 7-month improvement in PFS after switching treatment. These results emphasize that the importance of tracking ESR mutations in first-line endocrine therapy and support developing new oral agent to overcome resistance. Detecting mutation in ctDNA require high sensitivity and accuracy. The most studied method are dPCR and NGS, while advanced or the metastatic breast cancer often have sufficient ctDNA levels. High sensitivity technology like dPCR are essential for quickly detecting resistant-related mutations like ESR1. In the PADA-1 trial, dPCR was used for ESR1 mutation detection, with further studies showing how concordance with dPCR and NGS result. Next, Emerald trial was conducted to evaluate the efficacy and safety of elacestrant in patients with ER positive and HER2 negative metastatic breast cancer. And the result shows that elacestrant significantly improved PFS in patients resistant to existing endocrine therapies, particularly those with ESR mutations. Based on this result, elacestrant was approved by the FDA and the Europe commissions in the last year. Next, the recent AstraZeneca sponsored the Serena-6 trial is a ctDNA-guided early switches study evaluating switching to camizestrant plus CDK4 and 6 versus continuing with the AI plus CDK4 and 6 inhibitors during first-line treatment of HR positive, HER2 negative advanced breast cancer patient with detectable ESR gene mutations, but no clinical disease progressions. In this study, ESR1 mutations are also tested periodically, and the study design is such that the treatment is switched if the mutation is detected. The study is ongoing, and the results are expected to be published in the near future. Gentrix Droplex ESR1 mutation test kit is a product that can be used for these studies. The Droplex ESR1 mutation test kit is an investigational product for the detection of ESR1 mutations in FFP and plasma samples from breast cancer patients. It covers most of the ESR1 hotspot and is currently being prepared for clinical trials for domestic and international approval. It has recently been used in the fulvestrant clinical trials in Korea, and a paper on the product has been published. It is also being used in several studies in German. Finally, I will introduce Gentrix, a developer of digital PCR-based cancer diagnostic products, and highlight some of the key technologies and the new product under development. Gentrix was established in South Korea in 2011. Gentrix provides molecular diagnostic solutions across the cancer care continuum, including early cancer detection, breast cancer prognosis, and therapeutic companion diagnosis. Its main products include the Geneswell BCT product for post-operative prognosis of breast cancer patient. The Droplex line for companion diagnostic and prognosis by cancer type based on this PCR, and the EDX product for early diagnosis. Several products are licensed in Korea and Europe, and many are sold in Korea and overseas. Gentrix has entered into the strategic partnership with Bio-Rad for the development of molecular diagnostics, for the solidifying its commitment to advancing precision medicine in oncology. Through the partnership, Gentrix is currently recognized as a licensed diagnostic kit partner for Bio-Rad. We are developing a range of IVD kits compatible with the QX200 and QX600 platform. We are also a member of Cancer X project in the United States and a partner of the International Association for the Study of Lung Cancer. In the Korean market, Droplex is used by major Korean general hospitals such as Samsung Medical Center, Yonsei University Shincheon Severance Hospital, Seoul National University Bundang Hospital, and is currently setting up the testing platform at Korea University Group Hospital. These are the Droplex test lines. As shown in the table below, several products have been approved as IVD in Korea and CIVD in Europe and South and East Asia. Over the past 5 years, Droplex has been recognized for its technical differentiations and clinical utility in the field of clinical research and clinical diagnostics. We are currently developing NSCS panel products based on multiplex digital PCR and are working to obtain licensed and commercialize them in Korea, Europe, and the United States. The Droplex workflow is as follows. First, we prepare the sample and the reagent, mix them well, and generate Droplex and run the PCR. Then it only takes a day to read and result and receive the final report. When developing molecular diagnostic products, accuracy is crucial. Primer and probe design play a key role in enhancing accuracy, with specificity and efficiency being the top priorities. Primer and probes must be highly specific to the target gene and optimized for the maximum amplification efficiency. In PCR, probes monitor amplifications by binding to specific sequence, and quantum technology is used to control fluorescence signal, improving signal accuracy. There are two quantum technologies, single quantum and double quantum. Single quantum probes are simpler and cheaper, but have lower quantum efficiency, causing more background noise and reduced sensitivity. Double quantum probes with two quantums improve signal-to-noise ratio and enhance detection of low DNA concentration, but are more expensive. Single quantum is suitable for simple experiment, while the double quantum is preferred for amplification requiring higher sensitivity and accuracy, such as digital PCR. Here, I would like to describe our recently developed multiplexed digital PCR-based lung cancer panel test. The droplex cancer panel assay test is designed to identify a total of 180 hotspot mutations in 11 genes that need to be identified for treatment selection in NSCS patient FFP and plasma samples. The key point, it used multiplex digital PCR, and the test can detect gene alteration like SNVs, insertions, deletions, and also fusion, even from very small sample. It is particularly useful for patients with insufficient tissue samples and can monitor disease progressions to help guide the treatment decisions, and also benefit including reduced sample preparations time and cost, and patient examinations burden. To explain the sample process, DNA and RNA are extracted from the patient tissue or plasma sample. In the case of RNA, cDNA is synthesized. This type PCR is performed on the both DNA and cDNA simultaneously, and the results are analyzed. All of this can be done in a single day. We have recently presented our findings on the droplex cancer panel LC test as posters at Ask a Breakthrough and WCLC, and currently preparing to apply to the FDA in the United States and South Korea for approval as on CDIVD product. Thank you for listening to my presentation so far. In conclusions, I'd like to say that with the development of various digital PCR instruments, I expect that various cancer-related studies using these digital PCRs will be published in the future. I hope that my presentation will be useful for your research. Thank you.
Thank you, Dr. Hung, for that informative presentation. If you have a question and have not done so, please type it into the Q&A box. Okay, let me see. There are a few questions. So here we go. Here's the first one. Are there any special considerations or challenges when working with digital PCR compared to quantitative PCR?
Oh, that's a good question. Well, I don't think there are any major difficulties when it comes to preparing the experiment. But this the PCR is more sensitive. There are some things you need to pay attention to. For instance, it's essential to use positive and negative controls, and you will also need to establish appropriate cutoff value to ensure accurate result.
Okay, thank you. So next question. Why did you choose IDT over other companies for primers and probe production?
When we are developing our initial product, we tested the primers and probes from several companies. After comparing their accuracy, we found that IDT's products were the most reliable. So we decided to go with them.
Okay, well, thank you. And thank you for your business. Are there advantages and disadvantages of different digital PCR machines?
Yeah, definitely. Each instrument has its own pros and cons. For example, instrument A was the first one to hit the market. It's been widely used in bio basic research, and so there are plenty of references, which makes it easy to find evidence for its performance. But instrument B and C are more recent. They are a bit more user-friendly compared to the older machines, which could make them a better option for new users.
Excellent. Is there a reason digital PCR is a better option than NGS or next-gen sequencing for these applications?
Yeah, the advantage of digital PCRs over next generation sequencings are its cost effectiveness disadvantage and also fast turnaround time. And as I mentioned it in presentations, digital PCR can be done in 1 day, but NGS usually 2 and 3 weeks. So I think that this is a very good advantages to use digital PCR.
Excellent. Thank you. Thank you. So thank you very much for your answers. And unfortunately, that is all the time we have for questions. We are at the end of our webinar time. I want to thank all of you for attending today's presentation. I'd also like to thank Dr. Hung for her informative presentation and the Q&A session that we just had. If you have any other questions, please enter them into the Q&A box at the bottom of the screen. We will do our best to answer them through email or other means. As a reminder, we've recorded this webinar and will email to you a link to this recording. Additionally, this webinar will be available on the IDT website, where you can also find many other quality IDT webinars. Thank you again for attending, and we wish you the best of success in your research. Have a nice day.